computer. Okay. So Josh, without further ado, our Senior Vice President, Josh Bloom.
I reject that title. We're like, there's three of us. Like I don't need such power.
You were president, remember?
I couldn't get rid of that title fast enough. Anyhow,
He just doesn't want to wear a tie.
it was more like a noose, Jack, than a tie. I don't know. The day I talked Tom into taking that job was like the greatest day of my life. We were on.
You know, I didn't want it either.
Well,
the first words out of my mouth were, "I'm Tom Hagen, not Michael Corleone."
Understood. But on the other hand, we were on the 16th floor. I was fully prepared to turn myself into a 42nd Street pizza if you turned it down. So it, you know, it's a
well,
good.
As you know, I didn't commit right there. I said I wanted to talk to the donors first to get their opinion.
Anyhow, it's working out great. We're small, mean, lean, and well, all those other things. So anyhow, I kind of happen to be, I don't know, uniquely qualified. Sounds snooty, but I did 25 years in pharmaceutical research before ACSH hired me in 2010, which was the same year that Pfizer gave me the boot after they bought Wyeth. Not an uncommon occurrence. But about half that time I was doing work in antiviral drugs, both in HIV and hepatitis C, and a little bit in herpes. So it just kind of fell in my lap at a good time because there aren't really that many people who are writing about this stuff that actually understand it. And so I've been writing quite a bit, actually more scientific than I normally would, because there's a subset of my readers that actually will be able to understand and enjoy, I don't know, or get something or other out of it. So I only have, I don't know, 6 or 7 slides. So I left it short intentionally, just so we could have a conversation rather than a lecture. I just need to get back to my screen. Sorry. There we go. You have the slides in front of you, or do I need to share screen? Sorry?
I think you need to share the screen.
Oh, no. Well, that's not going to work.
I've got the thing that gives you the ability to do it turned on.
No. Hope you guys are patient.
Icon at the bottom.
Well, oh dear.
You got it?
No, it's the 2-screen problem.
Then just go to one screen.
Oh, share screen. And it's not letting me. Not sure what's going on. Just give me a second. If you don't have these things in front of you, it really isn't going to make any sense.
Right.
Okay, so there's the Zoom meeting, share screen. How's that look?
Nope.
Nope.
Are you seeing anything except my dumb face?
Just you, Josh.
All right, give me a sec. All right, this ought to work now.
Okay, I just sent everybody a copy of the PowerPoint.
A wise move. I think I'll have this. Okay.
There you go.
Yes.
There we go.
You see the slides?
Yep.
Now I do.
How's that?
Got it.
Got it.
Okay. God. I had no idea it was this brilliant. All right, so as you all well know, we're extremely fortunate to have a couple of antivirals to complement the vaccines, which are showing some signs of weaknesses, especially in preventing transmission. At the time that they first came out a year ago, nobody in the world, I'm guessing, would have even imagined Delta and Omicron and all these other things. This is unprecedented. Of course, there's never been a virus like this that's gone around the world. And so everything is new. So there really remains a need for antiviral drugs to complement the vaccines. And this is something that one of my colleagues, Professor Kathy Radke at University of Baltimore, Maryland in Baltimore, wrote about, I don't know, it must be going on 2 years ago. And it went into the Baltimore Sun, and it was that that drugs will probably be necessary, not just vaccines. And we either made a lucky guess or we knew something, but they're clearly both necessary now.
Well, at the time she was working on drugs too.
I'm sorry?
At the time she was working on drugs as well herself.
She's an expert in antiviral drug discovery. So yeah. I made a nice friend online, and we've written a number of op-eds together. And so what I want to do is just talk a little bit about the strategy of combating COVID as it works in both of these cases, because they're two very different drugs. They work differently. One works better than the other, and it's a little complicated. So I'm just going to go through it, and you guys stop me if there's anything that you need to discuss. I really prefer to discuss rather than lecture. So I accept rudeness for interrupting as a compliment. Okay, so within the last 3 weeks, the FDA has granted EUA to 2 drugs. The one on the left, molnupiravir, is a nucleoside drug from Merck, and the one on the right is Paxlovid, which is a protease inhibitor from Pfizer. The difference between the two is obviously the mechanism of action. They have very different chemical and pharmacological properties, very different antiviral properties. And Paxlovid was actually designed to inhibit SARS-1 protease, and it works very well for that. Now, SARS-1 turned into a non-problem because it fizzled out because it wasn't contagious. Fortunately, the protease domain of the current SARS is identical to that of the first one. So it's not the least bit surprising that Paxlovid, which is actually an analog of the earlier drug, but works about the same, that it should work in slowing viral replication by inhibiting the protease. But just before I go further, the name is confusing. Molnupiravir it doesn't get capitalized because it's a generic name. Merck does not yet have a brand name for this drug in the United States. Even more complicated, Paxlovid is a a combination of this antiviral, which I just call Norm, because it's really hard to pronounce, plus something from the HIV AIDS days called ritonavir. And the actual antiviral is Norm, but it is rapidly metabolized. So as has been done before with HIV AIDS, ritonavir is a powerful inhibitor of the metabolizing enzyme. So when you stick that in with Norm, you get Paxlovid, and it works like a charm. All righty. So we really have to go back to HIV AIDS days in the 80s to understand the how we got here, because it's although this is a novel invention, it's not a novel concept. So let's go back to the 1987, where AZT, the first approved drug for HIV AIDS, was hit the market. And there's some controversy about how well it worked, but basically if it extended life, the life of people who were infected, it wasn't by much, and it was very toxic. So it was a bad drug, but it was all that was available. So just to note, HIV was first reported in 1981. So it's 6 years before a lousy drug comes out, and people were dying and dying and dying. You'll note that the first 4 drugs, which were all nucleoside inhibitors, were repurposed from the 1960s, when they were mostly being developed as cancer drugs. The only exception to that is 3TC, which was approved in 1995. That was actually designed and first synthesized in 1988. It was designed to inhibit HIV specifically, and that turned out to be probably the best, or one of the best, of the nucleoside and NRTIs, nucleoside reverse transcription inhibitors. And you can see over here that how this thing works, and it's nothing novel. AZT has an azido group at this position in place of a hydroxy group, which you find in thymidine. So AZT gets incorporated into the DNA, and without the 3' hydroxy group, the chain cannot be extended. Works similar to how acyclovir works against herpes. So things really didn't change in terms of life expectancy until Roche came out with sequinavir in 1995. And this was the first HIV protease inhibitor. And as far as I know, the first protease inhibitor to be a drug of any kind. I could be wrong on that. You tell me. But you can see the molecule is very different. It doesn't look like a nucleoside. It's big, it's complicated. Pharmacologically, it looks like a mess, because you can just look at it and tell it's going to have absorption and metabolism problems. But nonetheless, it did, it started the downward trend in AIDS deaths. So if you look over here, 1995, sequinavir gets approved, and death rates in the United States started dropping. And they've continued to do so, because at least two or three dozen drugs have been made for HIV AIDS, which are much more powerful than that. So there is precedent for using a protease inhibitor approach to shut down a virus. This has been done before, but let me just give you, if you're not familiar with this, here's a quick cartoon on how viral proteases work. So the genetic information is fed into the ribosomes, and they're basically protein machines. And the ribosomes take the nucleobases and put them together and come out with one gigantic polyprotein, which is non-functional. It contains all of the necessary proteins to make new viruses, but as a single polyprotein, viruses are not able to use it. So the scissors represent cuts that are made by the proteases. So they're protease cutting proteins. And you can see that at this point, you get a number of smaller proteins. And these are the proteins that go to make up the structural and functional part of the new virions. So this is where your nucleocapsule comes from. It's where all the enzymes required for viral replication come from. So without protease, viruses can't work. The second time this was used was actually in hepatitis C area, and I spent, I don't know, must have been 10 years doing that kind of work. And let's just say it was met with limited success at the time, because so this is 22 years after hepatitis C was first identified. Sharing and Vertex at the same time came up with protease inhibitors. And you can see that they're complicated. They look much more like Paxlovid than they do molnupiravir. And this was a significant advance because these drugs provided a higher level of cure of hepatitis C, but they weren't great, and they had to be given along with interferon, which was part of the previous standard of care. And interferon is terrible. It doesn't work well, and the side effects are nightmarish. So even though these drugs were of use, and they did cure a number of people, everyone knew that a much better drug, Sovaldi, was coming down the pipe. And basically, doctors stopped prescribing the protease inhibitors in waiting for Sovaldi, which is obviously a nucleoside inhibitor. Here's the nucleoside. So by 2013, Gilead had put out Sovaldi on the market. That became the $800 pill that everybody was complaining about, but really provided the first reliable cure for hepatitis C without interferon and in a much shorter time period. So protease inhibitors worked. In this particular case, nucleoside drug was far better. Let's see.
Like,
People, first, I guess I should just opine for a moment. People complained bitterly about an $800 pill, and you had to take, what was it, 8 weeks of it? So it was
It was actually 12 weeks,
12
Josh.
weeks, yeah. So it was not, it was
Yeah.
$96,000 for a course. First of all, nobody paid that. That was just the number that Gilead put out. And second of all, the old regimen of interferon with ribavirin cost almost as much, and you had to take it for a year and a half, and you're sick, and you didn't get cured. So I argued in an article that Sovaldi was actually a really good deal at $800 a pill. It's all perspective. So these poor guys sharing, I think Boceprevir sold about 2 pills before they pulled it off. Telaprevir had maybe 4. I don't know. Telaprevir had 1 good year with a $2 billion sales because it was better. But neither one lasted very long. Neither one is even available today because the hepatitis C drugs are far better than even Sovaldi. And now the cure rates are 95 to 100% with even shorter treatment periods. So, I mean, that was probably organic medicinal chemistry at its best. The work that drug companies did to, to use a mechanistic-based approach to come up with rational inhibitors and solve a problem. So HIV, hepatitis C, both brilliant approaches, although since hepatitis C did follow the HIV model, you could argue that HIV was a more brilliant approach. So, a matter of opinion. So there are actually 2 proteases in COVID. One of them is relatively unimportant, and the other one is important. And it's called MPRO. This is the main protease, a clever abbreviation indeed. So it's the main protease that if you inhibit it, hold on one second. We are at the halfway point. Excellent, because I'm halfway done. Thank you, Tom. It's worth mentioning really the scientific brilliance that went into how Paxlovid works. Here's a part of the molecule that I just call stuff. And this, it's not really stuff. This is what binds to the protease enzyme. But the active species is this nitrile here. There's a green star on indicating the carbon. And since the main protease is a cysteine type inhibitor, it's got a nucleophilic sulfur. This adds to the nitrile and forms a reversible covalent adduct. And that is a spectacular way to inhibit enzymes. You get it stuck on there, it comes off very slowly, and it really kills the activity of the enzyme. So all of this came out of work 2 decades ago to deal with the original SARS. And it's an incredible success story by any measure. On the other hand, there's molnupiravir, which works by a very different mechanism. You can see that its active species is called anhydroxycytidine, and you can see the small difference at the top of the molecule. So this goes into RNA, the viral RNA, by the normal method, and either fits in for cytidine or uridine. Once it's incorporated, a few nucleobases later, the virus begins to undergo error-catastrophe. In other words, it's so mutated that you have a bunch of non-functioning, partially formed viruses. So that's how molnupiravir works. It's an inhibitor of the RNA viral polymerase. Again, something you've seen in HIV and in hepatitis C. So here are the problems. In the original clinical trial, Merck reported 50% reduction in hospitalization, but that was an interim result because they saw positive numbers. They stopped the trial, which you should, with a result like that. Unfortunately, when they analyze all the final numbers, that dropped to 30%. And I think the 30% is why it's staying on the shelves in CVS. The good news is the adverse effects were actually higher for the placebo, which suggests that the pill made people feel better rather than worse. And you can see that 3 times as many people withdrew from the placebo group than from the trial group. So, it does work. Good news is it, as you'd expect, since it has nothing to do with the spike proteins, it is active against all the variants, and I would expect that to continue. But there is an FDA warning since it's mutagenic in a number of tests. So you don't want it going into a pregnant woman because then you could see fetal damage. You don't want breastfeeding women to be taking the drug. So that's a warning that has to be heeded. That said, for other people, a mild mutagen over a 5-day course wouldn't concern me in the least. You don't get lung cancer smoking 5 cigarettes for 5 days, so this doesn't worry me. And I think my final slide, is Paxlovid, which is wonderful but unattainable. And the clinical trial that Pfizer ran was gold standard, randomized, double-blinded, adults with at least one underlying medical condition that would make them susceptible to hospitalization or worse, and confirmed COVID. So knowing that the pharmacokinetics of the antiviral were poor, it was combined with ritonavir to boost the blood levels of the antiviral. And when it was given to patients within the first 5 days of symptoms, the results were really nothing short of incredible. In the drug-treated group, 3 out of 389 people required hospitalization. Nobody died. In the control group, those numbers were 27 and 7. So that comes out to a 87% reduction in hospitalization and a 100% reduction in death. I believe these were non-vaccinated people, so one can only imagine how well it would work in vaccinated people. Once again, the adverse effects were not a factor. The people who were getting the drug felt better. I'm sorry, let me just the downside of Paxlovid is that the ritonavir, which acts like grapefruit juice, basically inhibits one of the important drug metabolizing CYP enzymes, CYP3A. So you need it to boost the antiviral. On the same, but on the other hand, you have to be careful because it will also boost the levels of other common drugs like statins, blood pressure drugs, antidepressants, sedatives. So there needs to be some care paid during the 5-day period during which people will be taking Paxlovid. They may have to discontinue their statins or cut the dose in half. This is the kind of thing that doctors and pharmacists can handle. This is well known. But that is that is a concern. The other concern is how do you get it? And basically under the FDA guidelines now it's impossible to get because you have to be high risk, you have to have a positive test, and you have to take it within 5 days. Who determines what high risk is? Your doctor? So maybe not a walk-in clinic, no way. How are the positive results determined? There's a 4-day waiting period for PCR tests now. So right away, a PCR test immediately disqualifies you from using the drug. The home test kits you can't even buy, and they're less reliable. So this should resolve once the supply problem resolves. And just to blow my own horn a little, I did mention that this was from July 2020, and this was our op-ed in the Baltimore Sun. And, this is the blind squirrel nut thing. We got this one right.
What,
And those are my 10 slides. And I'll be happy to answer questions or whatever you want.
take it down?
Is the,
You want to take the slides down or you want to leave them up?
oh, well, you know what?
They all have them now. I sent them the PowerPoint too.
All right, well, I'm gonna put them back up from. All right, you should be able to see them. Just
What, you talked about supply problem. Is there a storage problem with either of the?
no, it's a pill. It's
That's
probably good for 100 years. The supply problem, it's a very bad synthesis. I've talked to some people who do scale up work for drug companies, and they're not allowed to say anything, but they've called it a bitch. But, Pfizer's a giant company. They just invested $1 billion in manufacturing facilities, and they will make enough. Probably in a few months, there'll be plenty to get.
Yeah, the challenge, I think, Josh, is with, there's a long lead time, right, from when they get starting materials to when they get final product. And so a lot of the starting materials, they've got to source large quantities, and, that all takes time to put together. So, and, one of the challenges right now, I think if Paxlovid had come into the medical arena when we were dealing with Delta, you'd have seen a lot of utility, but now we're overwhelmed with Omicron, and basically, as you say, the challenge is getting tested, right? There's hardly any tests. I'm seeing daily newsreels showing, testing facilities with cars going back miles, and it just seems that if you can't get a test, you can't get Paxlovid, and if you can't get Paxlovid, then you're just going to have to deal with whatever comes along.
I disagree, because I believe the Daily News has just accepted an op-ed from me and Dr. Jeff Singer. We often write together. He's Cato Institute, and we work closely together, talking about getting rid of the test entirely and letting physicians make the call on whether the drug should be prescribed. The testing,
Bravo.
sorry,
Bravo.
yeah,
Bravo.
the testing is ridiculous. The pre-existing conditions are ridiculous. They go from smoking to heart disease, which can be mild or severe, autism, neurological conditions. None of it makes any sense. So the only way to do this is to have your doctor decide who gets the damn drug and who doesn't. And the FDA guidance needs to be thrown out entirely.
Right.
And I'm hoping that will be coming out this week.
I think there's a parallel model with influenza and Tamiflu, and the only way to make Tamiflu work is really to take it early. And so you pretty much have to have it sitting on your bathroom shelf.
Of course.
And you decide when you've got flu.
Right.
Take it.
I believe I'd rather have flu than take Tamiflu.
Yeah.
Because it's a god-awful drug. It doesn't work very well, and it makes you really sick. I once wrote an article about the 4 worst drugs that are available, and I included Tamiflu. And I put a bunch of quotes in from people, and you would think it was cyanide the way they were talking about it. So I don't think this is safe and effective. Tamiflu is really neither. So I don't really think that's a great model. But the concept behind it is keep it in the medicine cabinet. If you feel sick, test yourself and take it. It's the only way for this to work.
But you said it's tough to find.
Now, another 2 or 3 months after I'm already dead, I'm sure it will be, I'm sure it will be available. I've got bad asthma, so I'm only partly kidding. So I'm in a pretty high risk group and I'm old. So the things I've been trying to get my hands on a bottle of Paxlovid over the last 24 hours, you wouldn't even believe it. But you can read about it in my article that's coming out tomorrow.
Which,
I have a
which,
question. I have
Sure.
a question. Hi, guys. Did you hear me?
Yep.
Can you hear me?
Yes.
Yeah. Oh, okay, sorry. So I have a question about how specific is this Paxlovid for COVID? Or is it something that any RNA virus would be sensitive to?
It is completely specific
Oh,
to COVID because the protease domain of SARS-1 and this are unique.
okay.
So you're not going to find this. I mean, you could presumably test it against a panel of viruses, and I'd be willing to bet you wouldn't find substantial activity against any of them.
Okay, great. I'm glad I asked that question.
It's a good
Thank you.
question.
So-
More good questions are welcome.
would it work with MERS though, or some of the other?
Almost certainly not.
Okay.
This thing was designed with computer-aided drug design to fit the SARS-1 protease binding domain. And I should have put a picture of it up. It's actually really cool, the way it sits in there. And you can actually see that the sulfur of the cysteine active site with a covalent bond to the nitrile that I mentioned. And it's brilliant. I mean, this is as good as drug design gets. It's incredible.
So could the same design be used to cure the common cold?
It's a good question. I don't think anyone really wants to bother. And there's, the common cold, there are 100 different pathogens that cause it or something. So I don't see that as doable. I mean, we're talking about a specific virus here, which at least fortunately for now has a conserved protease domain. So it's a viable target. I don't think you can do that with a cold at all.
So you probably couldn't do it for any of the flus either.
Well, the only flu drugs are inhibitors of entry to the cell. No, that's the Tamiflu, and the other one that's even worse, which I forget the name. I don't know of any that work inside. Mitch, you may know better than I. I don't think there are any that work inside the flu virus to disrupt the machinery of reproduction.
Yeah, there's no protease inhibitors, and in fact, I mean, the way the flu virus works is different because it's got a segmented genome, so each protein comes on a separate piece of RNA.
Yeah, I remember.
So you don't
that.
have the
Yep.
clipping.
Yep. Oh,
Okay.
I totally forgot about that.
Yep.
I'm glad you're here.
Thank you.
Josh, I'm concerned about our ramming some of these things through quickly. I'm afraid that the Chicken Little media is going to grab a hold of some negative reaction and call it the next thalidomide, and then we're going to end up with a whole bunch of more stupid regulations.
I can't say that won't happen.
Uh-huh.
I mean, the media can falsely scare you about almost everything. In fact, a couple of weeks ago, NBC wrote a hit piece on Paxlovid. Now, the article was COVID drug risky. Okay, fine. There's your headline. Then in the article, they actually went into detail and they said, look, this drug works well, it's manageable. They talked to people that actually knew they were talking about, but the headline pissed me off. So I went after NBC big time for that. And then, of course, once they did that, 100 other news sources picked up the same headline. So of course it's going to happen because we're a country full of idiots. That's my professional opinion.
Josh, it was all about the ritonavir issue as well.
Of course.
In terms of the interactions, and there was nothing about the problems with the drug itself. And as you said, all of that's manageable, and we've been doing it for years in the HIV space. I think what was interesting, I went to the FDA. I zoomed in to the FDA advisory meeting on molnupiravir, and that one was actually filed with the FDA prior to Paxlovid.
Yeah, I know.
But they ended up approving Paxlovid first, and then in the molnupiravir label, they actually said, don't use molnupiravir unless nothing else is available.
Yeah, well, it was a 13 to 10 expert panel drug. That's pretty.
Right. It's right in the middle.
That's not, one or two guys fall asleep and then it's rejected. So
Yeah.
I don't think it should have. You know what? I guess it should have been approved and then pulled. But it doesn't matter. No one's buying it.
Nobody's using it. Yeah.
Right. This is like, turds for sale, 3 for $1, and no one buys them because the price doesn't matter.
Yeah.
You can't believe the number of drugstores I've called, waited for an hour to really bad music just to get to the pharmacist and find out whether they have stock or not. I found one place.
Which pharmacy actually had it?
A place in Rocky Point, Long Island, had 26 doses out of the 60 that they were allotted last night. This morning it was 10.
Wow.
You better believe it's 0 now. So right now there isn't a single dose of it in Long Island or New York City. And there's a HHS site, which is really helpful in real-time availability of these drugs. And it's hysterical because you go to CVS in X town and you'll see 0 out of 60 Paxlovid, 229 out of 230 molnupiravir. The FDA approval is almost irrelevant because no one wants it.
What's the price?
Free.
Yeah, I guess they're free. I mean,
Yeah, they're paid for by the federal government.
yeah.
Oh, I didn't
And-
realize that.
Well, they should be in this case, until we get out from this latest mess. You can't be worrying about people having to pay for this. Because I'm not sure where you guys are, but the hospitals are a mess here.
Well, here the Maryland, Virginia, and DC are all reporting reduced number of new cases. So the case level's going down. Only one hospital in Virginia reached peak, but they still had their overflow from 2020. So they haven't really run out of beds. What the hospitals are complaining about is not having staff.
That is why the hospital.
Because they're testing positive, they can't go in. And so they're actually asking if hospital staff that test positive can mask up and still come in.
Some hospitals are requiring it now.
Yeah.
So if that
that's,
does,
they not, for the most part, no hospital in Maryland, Virginia, or DC reached 70% bed capacity.
Same out here in San Diego. The ICUs are not overwhelmed. The emergency rooms are overwhelmed because everybody with a sniffle or a cough and can't get a test runs to the emergency room for a test.
Yeah.
Hospitalizations are up and climbing. but people are not nearly as sick as they were a year ago.
Right.
We haven't reached our hospitalization peak yet. I'm intrigued by your editorial coming out about too much emphasis on testing, Josh. I think you're hitting the nail on the head. I have, as you know, I have a daughter there in Brooklyn. My God, she spent a fortune on testing, but it's all to keep her young 11-year-old in school, hopefully. Josh, is there room for more optimism here? You know that I like to look at things from a historical context, and I've written a little bit about the vagaries of the FDA, but we can't forget that it took a century to learn everything about polio and a decade to learn about HIV. And look what we have learned in this last 2 years, going on 3. How many other drugs do those great big companies have in trial right now? I bet you there are other trials going, and you've given us the context of the HIV and Hep C drugs. Wait another year and there's a better drug. So Paxlovid probably is not going to be the last candidate for
Don't
treatment,
bet
is
on
it?
that one, Jack. Keep in mind, the drugs for HIV and hepatitis C, 1 to 2 decades.
Yeah.
All right. The only reason Paxlovid is available is because it was worked on 2 decades ago on essentially the identical virus. We would not have it. As far as what's in clinical trials now, as far as I know, 0.
Really?
Yeah, the only other drug that was in clinical trials was some piece of garbage by Roche, and it did absolutely nothing. So if there's anything else in trials now, I am unaware of it.
Okay.
There was a
Josh,
tremendous, I'm
was
sorry.
that the one from originally from Atia?
Yes.
Yeah, that's the only other one I know
Yeah,
about.
it was right up there with Flintstone's vitamins
Yeah, yeah.
in terms of efficacy, except it didn't taste as good. Yeah, I don't see anything coming out soon. It's virtually impossible because everybody was so naive about the drug repurposing, and we got nothing except maybe fluvoxamine, because that's not how it works. You treat viruses with antivirals, not drugs for this, that, or the other thing. That's just the way life works. So Professor Radke and I have written a bunch of times that we thought, with the exception of testing other antivirals, that the whole idea of testing repurposed drugs was a complete waste of time, as it's turned out to be. Of course, there's still the ivermectin people. They dislike me, but I don't even really talk to them anymore. I mean, take it, I don't care. I mean, it's fine.
Any other questions?
Yeah, I wondered if you could share your commentaries that get published to maybe our email list, because I'm where I don't really have access to those papers very often. And
Well, we usually when Josh publishes something in a newspaper, it goes up on our website the day after.
oh, okay.
We give them a 24-hour period. And what it usually does, though, is Josh will just write a brief paragraph what the article's about, and then link to the original article back at the newspaper. But he's done that
That'll
several
work.
times.
I don't check in every day, but I check in a lot.
Well, it will be very little effort for me to simply take this email list, and assuming it does come out in the Daily News, which is probably 98%, just to mail it to you.
That'd be cool. Thank you.
Don't send him spam, Josh.
I live to serve.
Anyone else? All right. Well, thank you all
Thank you.
for your support, and thank you for participating in today's Jonas Salk Society meeting. And we will be scheduling one for sometime in February.
Great.
The
Let me thank
speaker
you all
has-
too, because there were some great questions and some information I didn't have.
Correct.
So this was, certainly as useful for me as it might have been for you guys. So
Yeah.
I'm very glad I did it.
Great.
Thanks very much, Josh.
Very
Okay,
great.
Mitch.
Thank you all. Take care.
Bye-bye.
Bye-bye, everybody.
Bye.
Bye.
Thanks, Josh.
Bye. No problem.